Thanks for joining today. So my name is Gina Rajan, and as Flaminia said, I'm a Fair Data Coordinator at EMBL-EBI. And today I'm going to talk about open fair data and the role of BioSamples as a public data archive. And my colleague Ibuke will follow up with BioImage Archive, another public repository. So first of all, apologies to those of you who already know what fair data is. So fair data is findable, accessible, interoperable, reusable. And this is very much about, making your data findable. How can people find your data? Making it accessible, how to access and point people to where they can download the data, making it interoperable. So making it available to be analyzed by both machine and humans, and making this reusable. So ensuring that it can be integrated with other similar data using kind of specific standards, etc. So why do we want to have fair data? Why is it so important? A number of years ago, the European Commission, they commissioned management consultants to come in and looked at the cost-benefit analysis of fair research data in the EU. And so the result was they reckoned the likely cost of not having fair research data was about 10.2 billion euro per year. And they based this on kind of time spent generating data and research duplication and looking for data as well. And, fair data is really important as we kind of move on, and, data and data sciences obviously are core elements of research and innovation. And we all know there's kind of the growing impact of AI and machine learning, and this all requires data that computers can understand. And currently, many of our data generation efforts just don't support this level of reuse. And this Forbes article from 2016 is quite old, but it kind of looked at what data scientists spend most of their time doing. And it turns out 80% of data scientists' time spent collecting and then cleaning data, and it is the kind of part of their job they like the least as well. So how can we go about making our data fair? Well, in order to make it findable, we want to ensure there are persistent identifiers. So these IDs will never change, and they're associated with the data. We want to ensure rich metadata, and I'll talk more about this later. So metadata is the data around the data. It describes the data. We want to have indexed data repositories to be able to find the data again. And it's very important that the metadata also has persistent identifiers. To ensure the data is accessible, we want to have standard communications protocols so people know how to get it. And where possible, we would like these to be open and free. Sometimes authentication is necessary, particularly around clinical or human data. And we want to ensure the metadata is always available. So even if the data is retracted for whatever reason, there should always be the metadata should remain, because it gives that provenance of what happened to that data. We want to ensure our data is interoperable. We want to use vocabularies to describe them. And we want to ensure these vocabularies are fair as well. And having linked metadata is very important. And again, I will explain this later. To ensure our data is reusable, we want to ensure our metadata have multiple attributes. It gives as much information as possible. We want to have a usage license so people know how they can use that data. We want to have this provenance so people know where that data has come from, where it's originated from, what has been done to it before they come to use it. And we want to align with community standards. If data is described in the same way in different projects, then it can be used together. So just to reiterate the importance of metadata, it gives that context to the data. It supports the data interpretation. It improves data reusability and reproducibility. And it can be a very effective tool to find and access the associated data. And it's really useful to think about the research questions you're asking in your projects, and to think about the metadata that's required at the start of the project, If you kind of do this towards the end of the project, then you may have missed that chance to collect it. So this brings me on then to BioSamples. So BioSamples is the central EBI repository for metadata storage. It connects to multiple other EBI resources. And it's a core Elixir deposition database as well, so it's quite stable. It holds about 60 million sample records. And I'll just explain this kind of layer cake diagram. So built in these technical pillars, we have these BioSamples has these verification processes. So these are things like checklists that encourage submission of metadata via ontologies and controlled values. Many external projects use BioSamples as kind of a data management tool. They submit their samples and use it as kind of tracking for their data. And again, I'll explain this later on. And then BioSamples is a single hosting place for sample metadata, but it can link out then to assays and publications and also to external resources as well. So if you go kind of right back and look at this in a more granular way. So at the center here, we have a seawater. So we take this marine sample and thinking about the sample metadata that we may want to collect around this. We may want to collect sample metadata, excuse me, around the environment, the location where that sample was collected, the latitude, the longitude, the country, or the sea or ocean, the salinity of the water, the temperature of the water. And then you might want to collect metadata around the sample collection itself, the date on which the sample was collected, the time point at which it was collected, the actual tubes that were used to collect it. Sample preservation, you might want to collect things like the preservative used, temperature, and then for sample storage and transport, the duration of that transport, because that may affect the sample, and the conditions it was transported under, the humidity, the temperature, etc. And then for sample processing, you may want to collect things like the protocol, the reagents used, for sample isolation, and maybe the extraction method. And then for the sample storage, you might want to record the freezer temperature, the cryoprotectant used to store your samples. So all these things, it depends what it is you're looking at and how you think these things may affect your sample and therefore your data. So it's really important to standardize our metadata. And we can do this by the use of controlled vocabularies and ontologies. So this ensures this capture of metadata in a consistent manner. So BioSamples offers a number of different checklists. Some of these are the Genome Standards Consortium, MIXS. So MIXS stands for Minimum Information About Any Sequence. So these are around various different domains, such as soil, water, plant. And these checklists, these GSC MIXS checklists have many common attributes, but they also have specific attributes related to the domain. So for soil and water, there will be different attributes as well. There are also other attributes, but the Genome Standards Consortium makes up the most checklists that we have. And the Genome Standards Consortium is a working body that works kind of with communities to come up with these standards. The attributes in the checklists fall into three categories: mandatory, recommended, and optional. Mandatory means it is required that your submission will not get validated without that attribute in it. Recommended means it's quite a useful one to have in there, and but it's not mandatory. And optional is nice to have. And you can also add user-defined fields. So many of our projects that use BioSamples use user-defined fields. It's important to kind of harmonize these across the project, different partners in the same consortia, for example, before adding them. But this is a way to add kind of your own custom fields. So this is an example of a couple of the checklists. One is the Tara Oceans, which again is which is kind of project. This was done a number of years ago. The one on the right is the Genome Standards Consortium MIXS Water one. And you can see the The dark green are the mandatory attributes, the light green are the recommended, and the white ones are optional. So looking at BioSamples, I mentioned it connects to many other EBI resources. So it connects to metabolites for metabolomics data, FRIED for proteomics data, ArrayExpress for functional genomics data, European-C for publication data, BioImage Archive for image data, European Nucleotide for non-human genomic data, and European Genome Phenome Archive for human genome data, genomic data, and European Variation Archive for variation data. It also connects to BioStudies, which is a repository that takes in data that doesn't, it's linked to some of these other archives, but it also accepts data that doesn't fit into any of these. So the common thing here is the sample, because all of these repositories will ultimately have a sample link to that initial sample that you get out of the ground or out of the sea, and that metadata. So these resources will all have their own specific metadata as well that's relevant to the data type that they're collecting. So this is a screenshot of the BioSamples homepage. The link is there at the bottom. There is a search bar at the top, and you can search based on organism, environment, disease, etc. It covers a lot of domains. You can do a Boolean search. You can use a wildcard. There's also highlighted in the red there, there's a search button, and this brings up kind of this faceted search display, and there are filters, as you can see on the left here. So I've searched for marine metagenomes collected at a certain depth on a particular date, and as you can see, this has brought up 98 samples. If I click into one of these, I get this page, and this SAMEA followed by the string of numbers is the BioSamples persistent identifier. So this is what gets reused when this data is linked with other resources as well. It's referenced basically, and it can be referenced in external repositories as well and then connected. And something BioSamples supports is what we call relationships. So if you collect, say, a soil sample, and then you divide that up into 10 subsamples, these relationships are, these relationships are captured by BioSamples, as each of those 10 samples would have this derived from tag, and those sister 10 samples would have the same as. You may go ahead and do different analyses on all of those samples and link those to different data, but those relationships will be captured. There are a couple of ways to submit to BioSamples. So I put at the bottom here, again, the link to the production website. There's also a development website, which is very good if you want to do a test submission and just try it out. So there is a UI uploader. So this is our interactive submission. You can drag and drop a TSV file here. So you select your checklist. First of all, it's a list of checklists. You select the checklist you want to use. You upload your TSV, and then you get a receipt basically saying it's completed. And you can see all your submissions here, and you will get your accession returned as well. You can also submit programmatically using the API. So you, this is via JSON. So the minimum you need, if you're using the BioSamples minimum checklist, is just a identifier, so a name and the release date when the sample becomes public. So you can keep your samples private and set them to release in the future. And if you're using one of the checklists, then you would have to put in some of the attributes as key value pairs detailing these attributes, and you can add external links to related data sets, and you can add your relationships, publications, etc. And again, when you submit this, you'll get the response with the sample succession number. So I mentioned the use of checklists to submit. So we validate submissions to the checklist using BioValidator, which is an open source tool developed within our team. And this validates the metadata against the community defined standards. So, this is great for compliance with these checklists. So it supports JSON schema to validate the metadata structures and values. It works with different formats like JSON, XML, and TSV, so it's quite adaptable. And you can define custom validation rules with this. So the benefits of using this kind of tool is it improves the metadata quality and consistency, and it ensures compliance with standards. So just a recap on the biosample submission steps. The green are kind of the user input. So collecting this metadata template spreadsheet, and behind the scene, if you do it this way using the UI, it gets converted to JSON, or the submitter submits it as JSON, and then it gets validated and submitted, and the accession gets generated. And just to say, we have fairly good documentation if you want to have a look through this, if you're looking to submit or to search. And we have a help desk there, so please do email us. It's quite responsive. I'm going to go and talk about kind of how samples have been used for kind of data portals now. So I'm going to quickly mention a couple of use cases. First one is the Holofoods project, and this is the data structure for this. So Holofoods was around kind of host microbiota based on different feed types. So chicken, salmon, they were giving different feed types and looking at their microbiota and different data types on this. So here in the center, we have the chicken and the salmon, and then some kind of metadata around these animals and how they were kept. And then there were a number of derived samples, and these were associated with different data types. So kind of histology, inflammatory markers, genomes, kind of metagenomes, heavy metals, fatty acids. And you can see at the bottom, they were submitted to kind of biosamples, ENA, magnified from metagenomes, and metabolites for metabolomics data. So there's quite a lot of data there and samples. And what Holofoods did was they created a checklist for the metadata they were interested in. They used the relationships within biosamples to structure all those kind of samples in the way that they needed to. And then they made use of what we call structured tables. So structured for some of the other data, such as kind of heavy metals and fatty acids, there isn't a natural place to put this, but it was quite tabular data. So BioSamples supports the structured tables for tabular data that are associated with samples. And using kind of all of these sample relationships and this metadata, they kind of created this Holofoods data portal. So how this is built is quite a lightweight system. It's kind of a separate lightweight database, but it has the sample relationships and the metadata linking. And that's what kind of powers this. So you can search based on lots of things, the sample type, the animal type, and basically it brings you into that kind of gives you that holistic view of the data and access to that as well. And the publication for that is there on the right. I'm also going to talk about the biodiversity portal. So EBI has a biodiversity portal. This covers a number of biodiversity projects, some of them being Darwin Tree of Life, European Reference Genome, and Aquatic Symbiosis Project. So again, these use samples for their tracking system. So this tracking system is public. For some projects, they create this as a kind of for consortium use only, so project managers or partners can see where the data is at any point. In this case, this tracking is used to see if the metadata is submitted to BioSamples, if the raw data is submitted to ENA, if there's assemblies submitted, if there's annotation available. So this doesn't give access to the data, but it shows you kind of at what point the data is, at what stage it's being generated. And on top of that, there's then a data portal, which gives kind of some more search functionality and gives access to the actual data. So in many projects, the data portal is the end product. So it's that public facing interface to the data to enable search and access. And this is the Darwin Tree of Life portal. It has links to external references as well. And the main kind of point I want to make about this is, here we've got this really nice example within Darwin Tree of Life where we've got the sample metadata. This is linked to the sequence. And then here we have this link to BioImage Archive with the image of that specimen that the sample relates to, which is really nice. And as I mentioned, Ibuka will talk a little bit more about this. So just to kind of finish off, why do we want to use biosamples and why do we care about our sample metadata? So biosamples issues permanent accessions and enables metadata submission and, very importantly, validation around this for consistency. This in turn enables sample and data tracking and allows comparison of data across studies. BioSamples has this relationships functionality, so you can create these relationships between your samples. It links out to external repositories and also to publications, and it's a good way to feed into data portals and catalogs. And I'm going to end there, and these are our current and past team members and some of our funders that enable a lot of this work. Thank you.
Thanks a lot, Gina. And thank you for the great introduction to the FAIR concept as well. So I'll just pick up from there. Sorry, just clicking here. Yeah. Okay. So I'll be talking about the open FAIR data and how public archives can do it from the BioImage Archive point of view for your imaging data. So BioImage Archive is a relatively new archive. Our team leader and the first members of the team have started in 2021, but we are one of the fastest growing archives at EMBL-EBI, both in terms of the volume of the data. Obviously, the images are big. What we are doing is we store, index, and make available FAIR bioimage data, essentially like all the other public archives, to enable the reproducibility of the results, to enable new research, such as new analysis methodologies, to offer reference data sets, and obviously to enable reuse and to derive new results from the existing data. We operate across a very diverse domain. So these are this logo is done by the actual some of the images we have in the archive, just changing the color coding. And this is a random selection. As you can see, we have like this whole organism. This is a mouse, as well as down to subcellular structures and the images of those. So going back to FAIR, right? How do we enable this FAIR open data? How do we do the FAIR as the BioImage Archive? As Gina said, and or I assume all of you know, FAIR stands for Findable, Accessible, Interoperable, and Reusable. So let's start with findable, right? So as Gina said, you need the persistent identifier. So we have this, what we call accession ID for each submission, which is a persistent identifier. It never changes. And we also provide DOIs for your submissions. We also have the search functionality within the archive itself. For instance, here I've searched for brain, and it brings out the submissions which contains the word brain, as well as related terms such as hippocampus in this case. And so you can also search and find the images within the submissions. There is a search function within within the submissions, you can search the metadata of the images provided by the users in the search box there. How we do the accessibility, obviously we have this accessible, so like each submission has a web page. And as I said, through a persistent identifier. And also you can download both the metadata in terms of JSON and page tab formats, the submission metadata, as well as the images and related files, both via HTTP and also using FTP and Globus. Then comes the interoperable, which is the tricky part especially for imaging data, because this is just a screenshot, but we have in the biological imaging world, there are like more than 150 imaging formats, most of them proprietary. And especially proprietary ones, for instance, are not really interoperable. You need specific software, which, in most cases, for instance, is not free. And it's also due to these data format issues, you cannot actually visualize or analyze the data remotely. So you need to except for very small images in very specific formats like small PNGs, JPEGs, or TIFFs, you cannot really visualize or analyze the images without downloading. So this we have worked on and continue working on, and it is a longer topic. So I'll come back to this at the end of my talk. So bear that in mind. I'm not skipping it over, just leaving it for now for later. Then the reusability, which depends, as Gina mentioned, on all the previous parts, plus heavily on the metadata, because to be able to reuse something, you need to know what actually it is, right? So we, to describe what the images are, we have implemented the two metadata schemas, REMBI and MIFA. These are all coming from the biological community. So they were results of community meetings and workshops. And so we implemented these metadata schemas, which I'll just summarize in the next slide a bit more in detail, as well as using ontologies and controlled vocabularies where possible. So REMBI stands for Recommended Metadata for Biological Images. And this was came as a result of some workshops and published in Nature, I think, 2021. So it has 8 high-level modules. First one is the study, who are the authors, what is your project about, and anything like funding and publications. And then there is the study component, which actually corresponds to more or less the concept of datasets. And it's just a container of, what your images are about, what's the sample, specimen, how it's prepared, what's the organism, how the images were acquired, and if there are any correlative images, what is the correlation, the mapping, as well as if there is any image analysis applied, the metadata related to the image analysis. We also we don't only hold images, but we also accept and serve image annotations across all the scales of life, from whole organisms, as I said, to subcellular. We accept things like segmentations, masks, point annotations, class labels, and so on. These are like crucial parts of the data sets, especially if you want to work on new image analysis methods and appoint AI in producing new methodologies of doing these much faster and easier and more accurately. So for these, you also need to have some metadata, obviously, how these annotations were done. For that, we have held a workshop, I think now a year and a half ago as part of the AI for Life project. And out of the workshop came these recommendations. There is also a paper that is just published this year. And as part of those recommendations is the some standardized metadata for the annotations, like the which is called the MIFA metadata schema, like the annotation method, the type of the annotation, who did the annotation, was it a software, a specific version, or a human expert, or just any human, and so on. So we have implemented these at the BioImage Archive, in which you can see for each submission in varying details. In terms of publishing your data in the BioImage Archive, obviously, as any other EBI resource, we are a free public archive, so there is no cost. Size is typically not an issue. You can have huge data sets, and that is fine as long as they are not so they are associated with a publication or has a value in itself, such as a very commonly used training data set. We do accept that. You can control when the data becomes public, but we also provide reviewer access before the data becomes public. So you can share it with the reviewer of your publication or your colleagues. And then, as I said, we provide a permanent identifier as well as a DOI. The submission process is relatively easy and straightforward. You first check the scope. As I said, we need it to be associated with a publication. There are some, we don't accept any patient identifiable data. And there are some cases where, for instance, for volume electron microscopy data or cryo-ET data, where the data needs to go to the EMPIRE, which is another electron microscopy specific imaging archive at EBI. Then you just prepare your data as in, collect it into some folders, register an account, transfer your data, and create what we call file list, which I'll go into detail for the next steps, essentially, and then enter the metadata in the web submission tool and hit submit. In terms of transferring your data, you have multiple options. Again, we have the drag and drop option on the web submission tool, but this only obviously works for small images and low number of images. We provide the guidance on which to choose on our website. And then you can use for larger submissions, it's better to use FTP, Aspera, or Globus. And then, as I mentioned, we ask for something called file lists, which lists the files that are included in your submission. This would be one file list per study component. So if you have multiple data sets, then you would have multiple file lists. And file lists are also how we get the image level or file level metadata. Because we are a generalistic archive, and REMBI is a sort of generalistic schema for the biological imaging. There are some specific, metadata that changes from image to image or group of images to group of images that you may want to include in the file list. Again, for this, we provide guidance and examples, but there are no set restrictions because we do accept from many different modalities and experiments. But for instance, if you're working on some compound treatment experiments, then you can add the compound concentration, time, and so on into your file list as extra columns. And so add that metadata per image. Or if you're doing some genetic variation study, again, the gene identifier and so on. And then we have the submission tool where you enter the REMBI metadata and MIFA metadata if you're submitting annotations as well. So it looks like this. Basically, as I said, what's your organism, is if there are any extrinsic or intrinsic variables, how you prepared your sample. There are some required fields and optional fields. So and then there is you can see it in the page.json form as well in the new submission tool. And then once you submit, there is a submission web page, so a web page for each submission where you have all this metadata. So this is just a screenshot of the top part. There is more metadata relating to the sample and image acquisition and so on at the bottom. And then it also lists the files. Oh yeah, I've done the relevant screenshots. And then, so this is, for instance, a Tara Oceans submission. And I have a short video which shows what they included in their file list. So you can see the Tara IDs and also link to the Pangaea portal relating to the sample and so on. And as I mentioned before, you can do search in that file list as well, both as a general search, but also in all the different columns or multiple columns. This is another example, again, more connected to the biosamples. This is a submission done by Kopo at Earlham Institute for the European Reference Genome Atlas project. In this case, the images have their names both contain the BioSamples ID, but in the file list, we have a column that lists the BioSamples ID as well as a direct link to the BioSamples page for that sample. All the things that I have just talked about are on our website under the help and the metadata help tabs. As well as we have a help desk email, which is under About Us, Contact Us. We also have an online tutorial, which you can access anytime. And, you can either just do sections of it that is relevant for you or, do the whole thing on your own time. So everything I've showed up to now looks like this, right? So you go onto the submission webpage of any submission and you have some metadata, and then the images are like these boxes where you cannot see inside. They have some labels that describe what they are, like which sample, what kind of organism and how they were acquired, or like this additional metadata, what is the compound and so on, but you can't actually see. However, being an imaging archive, we want the users to have this experience of like visiting, similar to visiting an art gallery, essentially visiting a research art gallery, a scientific art gallery, so to say. So instead of the submission pages looking like this, they should look more like this, because visualizing images gives you quite a lot of information on top of what you can, get from the metadata alone. And there we go back to the I in the FAIR, the interoperability part. As I mentioned, there are these problems that we need to overcome, both to make things more interoperable, but also, as I said, to make them visualizable and analyzable on the fly, so without the need to download terabytes of data, essentially. So for this, there are some new generation file formats, NGFF formats, which does chunking and has a pyramidal structure. So chunking means all the data are stored in like chunks of arrays. And pyramidal is like you have different sort of resolutions of the image. So you have this original resolution, but you also have some lower resolution, smaller sized images in the file format in the same container, essentially. And when you want to view something, your browser asks for the relevant for instance, starts with if you're looking at the whole image, starts with the lowest resolution. And as you zoom in, just this part of this image, for instance, it asks for the relevant chunk of that image, so just this chunk, and shows this slightly higher resolution image, just like Google Earth or Google Satellite View, essentially. As you zoom in, you just see a smaller area of the image, but clearer and in more detail. So what we have been doing at the BioImage Archive, we accept more or less any data format, any image format, but we have been converting these into a new generation file format called OMESR. It's a standardized, community-supported, open data format, and it is cloud friendly because of this chunked and pyramidal structure. And this has enabled us both to standardize what we offer, but also to serve sort of a scientific image gallery. And we have, as a first step, we have done these galleries. We have 3 of them on our website, one for AI-ready data sets, just a few of them, not all of them we have, one visual gallery for select number of data sets that just to show the variety we have, and then a volume electron microscopy gallery for the data sets from Empire, again, just a few of them. This was like a first step towards a more before generalizing and applying this to everything. So I'll just show you a screenshot of going into the visual galleries. You can do this. So these are all online available. So this is a spatial transcriptomic data set, has 3 channels, and then 5 Z slices. And this is all online, and this is an image of 20,000 times 30,000 pixels. We have much larger images as well, and you can see, you can zoom in, change the intensity of the, visualization of the channel, and so on. I have another example. This is a time series looking at the calcium signals in the Arabidopsis, and you can see how the signal essentially is transmitted. And then last, I have a volume electron microscopy example, just to show you. We have links to different visualizers. Again, all of them are free open source ones. This is when you can visualize in 3D. For 3D, we also have NeuroGlancer links where you can see the orthogonal views. So on top of that galleries, the next step is to enable this fair image data in a browsable, accessible, reusable manner for all the data sets that are in the BioImage Archive. So the next step is from this to this that we have been working on. I think by the end of this year, we should have the beta version of our new website where you can browse the images. It will only be a few images per data set, but it will give the users a much, much better idea as to what they contain. So going back and recapping, as I said, we are a deposition database where we take the data which are associated with the publication, but it says on publication, but please do not wait until your paper is about to be published because, huge data sets take time to upload and get processed. with the associated metadata, and then we serve it. As I said, it can be used for methods development, training, or new discoveries, as well as by more specialized resources, such as data portals, like Gina has mentioned. And adding these will basically enrich the scientific value of the data sets and enrich the image data ecosystem. And going back to the data portals as to one of the examples of doing this, as Gina has already showed, this is an entry in the Darwin Tree of Life portal, which connects the BioSamples and BioImage Archive. But also, with, for instance, TREC, there will be a TREC data portal, which can connect even more archives and gives you the ability to visualize the images as well. And last but not least, I would like to acknowledge our growing team, which Matthew Hartley is our team member leader, and our wonderful growing team. And then before taking questions, I'll just show we are here at the just before the last in the webinar series, and there will be another one next week if you want to. And then all the other ones are accessible, recorded on the EBI training website. And yes, any questions?
Yeah, thank you so much, Ebuka and Gina, for the presentation. So we have a few questions in the Q&A. I ordered them from most upvoted, and there are two that I guess are connected, so maybe we can take them together for both speakers. So the first one is, are all metadata submissions submitted to BioSample manually, or has there been any tool developed over BioSample by any group, so biodiversity, All Food, Darwin Tree of Life, to automate submissions? Another person is asking, do you have standard templates for metadata in submission BioSamples and BioImage Archive?
So I'll answer the first one first about submissions submitted to BioSamples manually. So yes, large projects have developed tools to submit samples to BioSamples. So with individuals, usually with the TSV, you can still submit hundreds of samples. With JSON, you can submit thousands of samples. So Darwin Tree of Life use COPO to do their submissions. They submit the samples to BioSamples, sequencing data to ENA. So there are tools. It's something we've thought about doing as well. It's just a matter of, it's on the list, but it's a matter of resource for us. We don't recommend any tools specifically because we're not directly involved in those, but there are tools out there.
Thank you. And the second one, maybe I can repeat it just to refresh, was do you have standard templates for metadata in submission BioSamples and BioImage Archive?
So yes, the checklists are essentially these templates. They're available as kind of XML, JSON, or TSVs. So we have what's called a JSON schema store where all these are stored. I can put a link to these and to COPO in the chat, actually, where these are stored. You can also find many of the checklists in ENA where you can actually look at the attributes. We have them stored in JSON format, but you can convert to TSV and use that as your template and then upload that.
Yeah, similar with the BioImage Archive, we have, as I mentioned, REMBI and MIFA, and that information can be found on our website on the reference model. But in terms of so we encourage mostly the web submission tool at the moment. There are other ways to submit if you contact us for, like a programmatic submission. But currently, the BioImage Archive relies on the BioStudies submission tool because our focus was more on serving the images rather than the submission part until recently. But the BioStudies submission tool is very generalizable. That's why we don't encourage using their API because things can go wrong. But yeah, so in terms of the metadata submission, we are using the REMBI and MIFA templates and that information on what fields they have and examples and so on. Explanations are all on our website.
You. Okay, the next question is interesting. Someone is asking, as someone that is accessing the data on your public archives, how much freedom I have in what I do with those data. So for instance, can I develop and then patent a classification algorithm using the images for which I request access? Or is there some data access policy?
So
on this?
I think EBI has a fairly, so I don't want to say standard, but okay. So for BioImage Archive, we serve the data either under CC0, which is you can use for anything essentially, or CC BY 4.0, which is again you can use for anything, but you just need to give a reference to the source. So these are the two licenses we serve all the data in the BioImage Archive in. And obviously the submitters are aware, so you have only these two options when you're submitting your data to the BioImage Archive.
Right. So the answer would be like, yes, I can take those images and
use it. Yeah.
Okay, thank you. I don't know, Gina, there is something you want to add, or we can move on to the next question.
Also, all the biosamples data is freely accessible. I think that one was possibly about BioImage Archive. Thanks, Maria.
Perfect. Thank you. Then someone is asking if metadata are used only for usability or also for findability. So I think, can I query with metadata basically on the archive?
Absolutely. I mean, that's one of the reasons, especially if you're using standardized formats, you can search for that data using terms as well. So that's why you can use wildcards. So, the more standardized your data is, it's easier for people to find it.
Yeah, exactly.
Because,
sorry.
I was going to say, especially because we list what checklists it has been used to submit it. So, if you want to find other data within that data set, for example, then you know what you're querying for. And if it's a community standard, then you expect kind of all those similar projects to be using those same terms. So it makes it easy.
Yeah, and I would say that is one of the main things that basically distinguishes open data and open fair data, because if you just dump your data as a zip file in a Google Drive or somewhere, even Zenodo, and don't add the metadata, no one will be able to find it because there is nothing that indicates what that data is without the metadata.
Thank you for making this distinction, open and open fair data. I think it's important to re-stress it every time that there are two different things, or they can be different, let's say. Okay, next question is for BioSamples. So is the permanent accession provided by BioSamples an equivalent to an IGSN, International Generic Sample Number, when it comes to physical samples?
I would say no, because we use it's a similar concept, but we have synthetic samples, for example, as well. We have so it's not quite the same. Often it relates to a physical sample, but it doesn't have to. It can be some other kind of sample as well. It's what the user defines as a sample. So it doesn't have to be that physical sample. But a lot of the other concepts are similar.
Thank you. And the next question is it possible to change the images or metadata in the bioarchive once submitted?
So yes, you can change both metadata and add or delete images before it is released, before it's public. Once you submit, when you're sharing with your colleagues or reviewers. But once it is public, you can add images or change the metadata, but you cannot delete the images once it's public. That's the only distinction.
Okay, but if it's not public, they can delete images. Okay, thank you. Then we have a question is, can we have metadata for a particular genome or protein? So I'm not sure I'm
So I think
understanding.
this probably means more annotation than metadata, because I think this is referring to annotation possibly, and therefore you'd need to go to a genome archive or a protein archive to get that annotation. I think that's what that is referring to.
Yeah, yeah. Or like if you mean in terms of, edit, either for, I don't know, because biosamples have the connection to ENA, or like, do you mean in terms of the
Yes.
columns to the BioImage archive as well? If so, like if you are working on a particular protein or mark your images with a particular protein, or then you can add that as a column in your file list, for instance.
Yeah. So maybe what we can do to the person that asked this question, if you feel like we haven't replied, you can maybe just develop it. You can just type another question, and in the meantime, we can take the others. Okay, we have 2 questions left. So how can you research using this tool as a BS graduate? So I guess that's a Bachelor in Science. I'm not sure what BS acronym stands for in this case. So maybe if this doesn't ring a bell to the speakers, I would ask the person who wrote this question to specify what BS means in this case. It's Bachelor of Science, okay.
So I think it depends what you're using it for. If you want to play around with them and try them, we have a dev site, so you can try it out and see how to use these tools. So the things on the dev site never go live. And if you're actually working on a project, then with kind of data, image data, whatever, sequencing data and sample data, then you can try these things out, I guess. There's extensive documentation and tutorials on how to use these, so it's worth having a look around the website for that.
Yeah, I mean, both of these archives are generalistic archives, so they are not domain specific or, sort of method specific. So it all depends on what you have in mind as a research question. You can just go and find related samples, find related images, do something with them or check, whether that method works for the thing you have in mind. There are various reuse options. Just depends on what you want to do.
Thank you. So then we have a question for BioImage Archive. Oh, it's 3:30. Is it okay if we stay a couple minutes more and we take all the questions?
Yeah, no, it's fine for me.
Okay, thank you. So for BioImage Archive, are there quality checks of the uploaded images? So the person is saying, especially I'm thinking about AI-generated images. Could they be detected at the submission step and prevented from upload, or maybe add a disclaimer like this is AI-generated?
Yeah, that's a very, very good question. So we have very basic quality checks, but we don't. I mean, I think at the moment that distinction about AI-generated images is a quite difficult thing to do. We had discussed about it, thought, because we will definitely face a similar issue in the future. But we don't have anything in place at the moment, but that is definitely something in our minds as well.
Thank you. Then do you have a process in place? Oh, sorry, it moved because someone asked another question. Do you have a process in place to review whether the submitted images are de-identified, thus clean of protected health informations?
We don't do active checks or curation related to that. That is like, as if you really, really want to hide somewhere in the headers, we would come across it during the OMISAR conversion process, then, taking the necessary steps. But essentially, or if it is obvious in the metadata, obviously it will be code, but we don't have sort of a regular process that goes through that.
Thank you. So there are three more, so I would take those three and then conclude the webinar. So there is one that says, I've received some data from NCBI. Now can I know that we can do a comparative study with the related data available in BioSample? I'm not sure. So if you have the Q&A open, Gina, maybe you can also just read the text. Can we do a comparative study with so I think they wanted to compare between data that are in NCBI and something that is in BioSample.
One thing just to mention is if it's sequencing data, then it's mirrored between NCBI and ENA. And BioSamples is also part of this INSDC International Nucleotide Sequence Database Consortium. We mirror data across. So that sample you may find in NCBI and in BioSamples. If you're looking for related samples, then depending on what kind of metadata is used in that you have related to that data, you can do a search for that in BioSamples to see what other samples that brings up, which should lead you to potentially sequencing data if that's what it is you're looking at. I hope that answers your question.